afternoon, everyone, and welcome to another one of our virtual Research Cafe 10.0. I'm Frank Gomez, Executive Director of STEMnet, and it's really great to have you all here today. Behind the scenes, we have Monica Alacon, Administrative Analyst for STEMnet. Much thanks goes to her because she's, I guess, kind of the brains of the outfit who's really walks me through all the technology aspects of all of this. The goal of the cafe is to help foster collaborations among CSU faculty and to catalyze submission of proposals, the joint ones and multi-campus ones at that. It brings together assistant professors in the CSU to share their work in a relaxed setting and for 10 minutes each, reason for why we call it 10.0. We hope you'll find opportunities for collaboration here and learn about a new area of research that may impact your own program. Please feel free to contact any presenter in the chat box when they're not presenting. At the end of the 3 presentations, we'll bring everybody together to answer your questions. I'll also be monitoring the chat box to pull questions from the audience. So let's begin. Our first speaker is Dr. Yixian Wang from the Department of Chemistry at Cal State LA. So Yixian, the floor is yours.
Oh, thank you, Monica. Thank you, Frank, for your nice introduction. I'm going to share my screen. Hello, everyone. Thank you, Frank, again for your nice introduction and for inviting me here to present today. I'm very happy to have this opportunity to present my research to colleagues across the CSU system. I have to wear a mask because I'm in the lab with my student. So to avoid any contamination, I have to do this way. Again, thank you very much for being here today. My research interest mostly centers in single entity analysis. And when we talk about single entity analysis, I'm referring to single cells or single nanoparticles. So you're looking at 2 different scales. One is ranging from a few micrometers to a few tens of micrometers, and the other one is from a few tens of nanometers to hundreds of nanometers. We actually use a variety type of sensing and imaging techniques for single entity analysis, but today I will be focusing on plasmonic imaging. First, all the plasmonic imaging systems are based on surface plasmon resonance. You might be familiar with the traditional SPR technique. In a conventional SPR spectroscopy technique, the SPR utilized the collective excitation of electrons in a thin gold film, for example, a gold film. It can be used to probe small changes occurring at the solid solution interface. So typically it's used for study interaction between biomolecules. So for example, in the traditional setup, the detector will be able to measure the drift in the SPR resonance angle. And as a result, you can get a sensogram, which can monitor a molecular interaction, for example, antibody-antigen interaction. But in a word, you're getting a spectrum type of signal. Versus SPR microscopy or imaging, we are actually getting imaging capability. So we're still using SPR signal, but with the imaging capability. So for example, you can see here, you can monitor the binding process over a certain view, and you can see individual binding events simultaneously. Or if you're looking at a single cell, you actually can look at different portion of the cell. And by monitoring the changes at different position, you can get different sensogram at different positions simultaneously. So that's what's good about adding the imaging capability to SPR measurement. So this is one of the main techniques we use in our lab. And to add to that, SPR can also be combined with the electrochemical measurement. And we call it plasmonics-based electrochemical imaging. And this was first invented by late Professor Longjian Tao, who was also my postdoc PI. So in 2010, they first reported this work using SPR signal to measure the current going on in an electrochemical reaction. And as you can see here, the signal from the SPR measurement can fit with the actual cyclic voltammetry obtained of potential step very well. And later on, they applied this technique to study the hydrogen evolution at individual platinum nanoparticle. The example we're showing here is actually from a nanoparticle that is about 8 nanometer in size. So it has a very good sensitivity. And just to add a small note to the shape of the signal, when you're using SPR or plasmonic imaging technique to look at individual particles, you will normally see a very long tail behind the particle. So you can imagine that like a boat in the river because the surface plasma is actually a propagating wave, and it will be diffracted by the nanoparticle and leave a long tail behind it. And this is kind of good because it's easier for you to identify nanostructures in SPR versus a normal bright field. Okay, so this is a little bit background. And talking about the instrumentation in our lab, we have two setups for plasmonic imaging. The left one is kind of a handmade one. So it's a combination of different imaging and sensing techniques together. So it's a hybrid system. And the right one is a recently purchased instrument supported by an SFMRI grant. If you compare the two instruments in their SPR and the plasmonic imaging capability side, the handmade one tends to have a better spatial and temporal resolution, but the commercial one has a bigger imaging view. So you can image more entities within the view. And of course, the commercial one is more user-friendly. And it depends on what type of experiment we want to do. We choose to use different one. Then I will give you a few examples of the ongoing project we are doing in the lab. The first one is we would like to use this plasmonic-based electrochemical imaging technique for studying single nanoparticles. We have previously worked on demonstrating the capability of getting cyclic voltammetry at individual nanoparticle level. So here you can show that those two are the videos of showing that you can monitor the electrochemical changes at individual nanoparticles. And if you extract the signal from each tail of the nanoparticle, you can get the single nanoparticle CV. And if you have a nano wire, so in this case, we have a 10 micrometer gold nanowire, which has about 60 nanometer in cross dimension. And when you have a relatively long dimension, you can actually see the distribution of electrochemical activity within a single wire. So this is what we show here, that a sub-nanoparticle level detection with SPR, with plasmonic imaging. Now, a current ongoing project in my lab is we're interested in seeing how the crystal facets of individual particles are affecting their electrochemical or electrocatalytical activities of those particles. And to show you an example, we studied gold nanorods. And if you're looking at the CVs extracted from those single gold nanowires, you can see that there are some tiny minor peaks, which are actually can be contributed by the facets, the surface facets like 100 or 110. And another interesting thing is when we do different cycles, you can see that the position of the peaks are actually drifting from during all the 4 cycles, which could suggest that there is a morphological change within during the scan. So this is one of the directions where we're still working on. And I haven't mentioned that we're also interested in single cell analysis, and I like to call it selfie because we do a lot of imaging of individual cells. And I already talked about the SPRM part, which we use SPR to sense the binding or any changes occurring at the cells. We also have two scanning probe-based technique, imaging techniques, the AFM, the SICM. So simply saying that you're using a scanning probe to scan over sample surface to get topographical information or mechanical information or other information. And residual pulse is just another sensing technique. And we are collaborating with different groups. For example, we are collaborating with Dr. Edith Porter from microbiology to investigating the roughness change at individual bacteria. And ourselves, we're working on Parkinson's disease project, trying to investigate the effect on alpha-synuclein on the membrane disruption that can be related to the progression and development of Parkinson's disease. And we also have some ongoing project using SPR. On the Parkinson's disease project, we also have collaboration with Dr. Jamil Moment's group about the drug resistance development on cancer cells. So here shows the example that we can map the binding affinity at different cells simultaneously. And for that, I would like to conclude my talk by acknowledging my students, all the students who work very hard, especially Adelie, who is the only student that is allowed to go back to the lab now. I actually just took the photo today in the lab, and all the funding agencies to support the student and the research, and all my collaborators. I actually see two of my collaborators are actually here today. Thank you. So I would like to especially mention that we are really interested in looking for collaborators in neurodegenerative disease. We are more like an analytical chemistry group. And if you're interested in this type of research from a biochemistry side or a biology side, I would really grateful to establish a collaboration. But on the other hand, we are also willing to share our resources of all the instrument. If you would like to do any measurement imaging of binding activities or cell morphological changes during any bio process, please feel free to contact me. Or if you're interested in studying the heterogeneity of your nanomaterials, I think our platform is also a good platform to test that out. And thank you, everyone.
Thank you, Yixian, for a very, very interesting presentation. Our next speaker is Sudhir Shrestha from Sonoma State. And he is from the Department of Electrical Engineering. So Sudhir, I hope you'll be able to unmute yourself. Floor is yours.
Thank you, Frank. Let me share my screen here. Okay, right. Thank you very much. Again, my name is Sudhir Shrestha. I'm an assistant professor in the Department of Engineering Science at Sonoma State University. A little bit about my background. Let's see here, I can advance my slide. Okay, here we go. I completed my bachelor's degree from Kathmandu University in Nepal. And I got my PhD from Louisiana Tech University. And then I went to Indiana University, Prairie University in Indianapolis, IUPUI, for my postdoc. I continued there as an assistant research professor. And this is where the research I'm going to talk about began. Later, I went to Miami University as a visiting assistant professor. I joined Sonoma State in 2017. And besides, teaching and research, my wife and I love to hike and just wander around and get closer to nature. And that's the one of the greatest thing about Sonoma County, that you can do that easily and as often as you want. As part of my research, we work with sensors, collect data from the environment, and try to make sense of data using various tools, and use that to for solving problems. And we do that by designing hardware, software, and developing test and then various procedures for that. And currently, this work is focused on biomedical sensing, computer vision, and robotics. But today I'm going to talk about blood glucose monitoring, and this is about being able to monitor blood glucose level using breath. Diabetes is a huge problem in the United States and around the world, with more than 34 million people with diabetes, another 88 million people with prediabetes. And one of the scary stat in here that half of the people who have diabetes do not manage it very well, and that put them in the risk of complications, many health complications. So this is such a widespread problem that even a small contribution in this field can have a major impact. And one of the reasons we want to detect, develop diagnostic tool using breath is because it is non-invasive. It is something easy to do, and doesn't take an effort, doesn't involve any finger pricking or any kind of poking at all. So anytime there is a way to develop a diagnostic tool, I think there is no reason not to do that using breath, right? In our breath, and really the reason we are able to do that is because in our breath there are more than 2,000 different volatile organic compounds. Those are added in our exhaled breath, and these VOCs come from various sources. What we eat, and from our bacteria in our mouth, but also from our lungs, and various metabolic process in our body. And various health conditions are expressed as VOCs in our breath, and there is more than few decades of work in this area. A lot of groups around the world have done systematic work trying to correlate VOCs with various disease conditions, and diabetes was one of them. And there's a lot of clear established correlations there. And the bigger question is, all of these results are from controlled chemical studies in the lab. Can we use that to develop a diagnostic tool that people can actually use? So that's the question we're trying to answer. And before that, I want to share this information about diabetic alert dogs. So these are trained canines trained using breath and perspiration of patients when they are at a normal and hypo and hyperglycemic conditions. And there has been a lot of studies in this area, controlled studies with blinded studies with collected samples, and it has been shown that they can detect actually with more than 97% accuracy. So the initial thought process was, can we just mimic a dog's nose, right? So that was the initial thought process, and we set out to find what is in the breath that dogs detect. If we can find that out, we can make a sensor to target that, and we'll have a solution. And this is the work done at IUPUI, and you can find more information in this publication here. But as part of this, breath samples were collected from patients with type 1 diabetes. That was the thing we're focusing on. And looked into that using GCMS and analyzed the data. And what is found is there was clear, the group of VOCs had clear correlations between with various blood glucose states, and with more than 90% sensitivity and more than 80% specificity. And with that, we set out to make sensor arrays. We actually also developed our own sensors, but the results I'm going to share here are from off-the-shelf commercial sensors. So when you looked into some of the commercial sensors, we found really good sensors that can detect at the VOCs at the level we needed to. And so we studied those sensors and developed arrays. And then we created this system to create simulated artificial breath. It's basically ultra pure air with added humidity spiked with chemicals representing, normal high and blood, high and low blood glucose states. We tested our sensors using those. And you can see here one snapshot of results, but just one snapshot of result doesn't provide you much information. You have to look at the pattern of the data. And that's what we did next. We analyzed our data using a machine learning algorithm called support vector machines. And we implemented that trained algorithm in a microcontroller because we wanted to create a device. We tested that back with a simulated breath, and the accuracy was higher than 97%. So, but all of those tests so far and it has been with simulated breaths. So this kind of shows our workflow, and you can see, we started with the simulated breath and chemical sensor array, trained algorithms, putting that in the microcontroller, and developing a device. This you can show you one of the designs of the handheld device. And the next step here is to, do clinical studies for collecting data and also testing the device, and that's what we're on. So as part of the collaborations, if you are working on any kind of VOC sensors, our sensor still needs to be heated. So if there's any sensors that works with room temperature, that will be very interesting for us. And also we have a way established for clinical studies. We are planning for, we have an IRB for adult patients with type 2 diabetes, but we also want to expand that to younger demographic and also type 1 diabetes. So that's something you are doing, and that will be also interesting. And besides that, any part of the work that I talked about, there's so much problems that needs to be solved, and it's such a broad topic that there is a lot of opportunities for collaborations. I end by thanking all the other people involved here. Dr. Wilkos is a faculty member in nursing, and I'm working with her for the clinical studies, and all the students who have contributed greatly from sensor arrays to coming up to the clinical studies. And thank you, Frank and Monica, again for this opportunity, and thank you all for listening to my presentation and your time. Thank you.
Thanks, Sudhir. Great talk. I certainly have a few questions, but I'll wait till the end. Our next speaker is, last but not least, is Ornella Mattei from San Francisco State, from the Department of Mathematics. So Ornella, your turn.
Let me share my screen. Hello, everybody. Thank you so much for coming. Thank you, Frank, for the invitation, and thank you, Monica, for all the work behind the scenes. Before I tell you what I do, let me tell you a little bit about who I am. So I'm from, probably as you can tell from my name and from my accent, I am from a small town close to Milan. It's called Brescia. The only reason why you may know this town is if you're a soccer fan, because 3 of the major Italian soccer players, Baggio, Pirlo, and Balotelli, are from my hometown. So in Brescia is where I have completed my studies. I got my bachelor and master degree in civil engineering. And then I got my PhD in applied mathematics. But my PhD program was rather unusual in the sense that it was a combination of engineering and mathematics. So I'm not that comfortable like calling myself a mathematician, but I'm not even an engineer. I'm somewhere in between the two. After my PhD, then I moved to the States. I spent 3 years as a postdoc at the University of Utah. And that was like the perfect playground for me because I'm a huge hiker. I love being outdoors. And I concluded my postdoc last year, and I joined the Department of Mathematics at San Francisco State University. So you can imagine that it was a first crazy year for me. Okay, so what do? I'm mainly interested in all the problems that are related to the mathematics of material science. And currently I am focusing actively on 4 projects. The first one is wave propagation in time modulated materials. The second one is a bounds on the response of viscoelastic composites. And the last 2 concern inverse problems for time-dependent and time-independent inhomogeneous bodies. But today, I'm going to focus only on the first problem, wave propagation in time-modulated materials. So what are those materials? Well, roughly speaking, we call time-modulated materials all those materials that change their properties in time due to a change in the external applied field. As an example, you can consider liquid crystals. Liquid crystals change their refractive index due to a change in the external applied field. So these materials were studied since the 50s. So the first papers are from 1958. But then nobody studied them for a long, long time, mainly due to some technological issues in the implementation of these materials. Then suddenly in roughly 2010, we had a boom in the research of these materials for mainly 2 reasons. The first reason is that these materials can break reciprocity. The second reason is that they are the best candidates so far to create perfect absorbers. And the last reason is that they have a very exotic properties when it comes to cloaking. So for today, I'm just going to show you an example of a space-time material. So here we're working in the one-dimensional case. And you have a string that is made of alternating segments of material 1, material 2, material 1, material 2. As time goes by, nothing happens. And then at a certain moment of time, you change the external applied field. And what happens is that what before was material 1 now is material 2. And your material 2 turns into material 1. And then you keep applying this switch periodically. So what you end up with is what it's called a space-time checkerboard. So here notice that the vertical axis is time. So you see that you keep moving from material 1, material 2, material 1, material 2, and so on. And now the question is, how does a wave propagate through a material that has this behavior? I'm a mathematician still, so I had to put down one equation. Please forgive me for that. So this is the wave equation, and you see here that it's very, very simple. The only difficulty comes from the fact that the coefficients, alpha and beta, are actually functions of time and space. So how does a wave propagate for this material? Or in other words, what is the solution of this equation? The reason why we want to study this problem is that when you think about wave propagation, it's a time-dependent phenomenon. So if you want to realize a material that behaves in the optimal way towards a wave propagation, it has to change its properties in time as the wave propagates through. So if you fabricate the material without any specifics, then what you get is this. So let me explain what this is. In order to visualize the wave, we use these red lines that are called characteristic lines. And basically by tracing these lines, you have an idea about how the wave propagates in space as time goes by. So here you see that at the very beginning, at time t equals 0, the wave splits into 2 parts. One part that goes to the right and one part that goes to the left. And then you see that a mess happens. And the reason for that mess is that every time the wave hits an interface, it splits into 2. You have a wave that keeps going in the same direction and a wave that starts traveling in the opposite direction. This is what is classically known as a reflection and transmission. So the idea here is why don't we tune the switching in such a way that we don't end up with something so messy, but we end up with something that looks very nice. And the reason is that if here you want to track the wave after, I don't know, 100 switches, oof, that's going to be really hard, even if you want to tackle the problem from a computational point of view. So one of the things that I did during my postdoc was to work with the parameters and try to find combinations for which you end up with these beautiful patterns of characteristic lines that are called field patterns. And here, since everything is periodic in space and time, you just have to compute the solution once. And once you have it, that is keeps repeating in space and time because everything is periodic. So if you wonder how the solution looks like for this case, this is how it looks like. So the wave propagates through that triangular shape, and specifically it travels only along the edge of that triangular shape. And here, even far away from that edge, you still have oscillations. And this is something that does not happen with conventional materials, for which instead, after the wavefront has passed, you do not have any oscillations. And this is one of the exotic features of these materials. You can also do something fancier, and instead of the 2 materials, you can use 3 materials. And the reason why I'm showing you these case is that here, depending on how you pick the wave impedances that are the gammas that you see at the bottom of the slides, you can have 2 cases. You can either break PT symmetry, that stands for parity time symmetry, or you cannot break it. So in the first case, when the PT symmetry is unbroken, you end up with a solution that is roughly the same one that I showed you earlier. So the wave just propagates mainly at the edge of the triangle, but you still have oscillations far away from the wavefront. But if the PT symmetry is broken, this is what you end up with. This graph is in a logarithmic scale. So here what's happening is that the wave intensity is increasing exponentially in time. And at this point you may ask, wait, how is that possible that the wave increases exponentially in time? Well, think about what's going on. So here, in order to modulate the material, to do that switch at every period of time, we need to inject energy into the system. So at every time step, we pump energy into the system, and what happens is that the energy blows up. So one of the characteristics of this material is that conservation of energy doesn't hold. All right, so what am I doing right now? I'm working with some students at SF State, trying to understand how these materials behave if we assume that the interfaces, so the spatial interfaces between the 2 materials and the temporal interfaces are actually not sharp, but they are gradual. So that's why the first item is space-time functional graded materials. Then the second item is dissipative materials. And here the idea is, okay, we have a blow up of energy. Can we maybe counter that blow up of energy by assuming that the material dissipate energy as time goes by, and maybe we can counteract the 2. The third item instead is the characterization of the dispersion diagrams for these materials. If you're familiar with band diagrams, in general, you have horizontal band gaps, but for these materials, you have vertical band gaps as well. And this is something very unusual. Going forward, I'm really interested in collaborations actually in any field, but specifically going on, I would like to explore a little bit more how to break reciprocity and time reversal symmetry in the case of space-time checkerboards, but also how to add non-linearity, because here the problem is very simple. It's a linear problem. It's a one-dimensional one. So what happens when we extend these results to the 2- and 3-dimensional case? And this is it for my talk. I thank you so much for your attention, and that's it.
Thanks, Ornella. Very, very interesting. I certainly have a few questions too, but I don't want to, take over this. So we're now open for Q&A from the audience. You can speak up if you have questions for any of the presenters. By all means, just unmute yourself and go to it.
I have a question for Yixian. It's about your technique.
Also, if you can just announce who you are and say campus.
Sorry. Okay. I'm Dr. Call me Param. My name is, full name is Kodishwaran Parameshwaran. Feel free to call me Param. I am in California State University, Chico, and my research area is neuroscience. Particularly interested in studying the effect of abusive chemicals in brain development, such as nicotine. Nicotine is the major substance I study. So in simple terms, my research area is neuroscience. More specifically, I study developmental exposure to chemicals and how that affects brain development and cognition. My question to Yixian is, so for example, I'm interested in studying neuronal morphology. If a protein is lost due to a mutation, that might affect neuronal morphology. So using your technique, can we study that? And if so, do you use live cells, which I doubt that probably might not be the case, but I'm not sure. Is it preparations, fixed cells, or how it is usually done? So basically, my question is, what should be the preparation?
Thank you, Perrin. That's a great question. So we did use live cell, but they have to be immobilized on a platform, on a substrate. Normally what we do, the cell line we worked with is neuroblastoma cell, SHSY5Y. So we have to immobilize the cells on a small petri dish, and then we can image them when they're still alive. When my student was trying it, I think the cells can be alive for up to 10 hours during the imaging. And also the imaging will tell you when they started to die, because the shape will change during the imaging. But it's definitely possible. I don't know how the viability for primary neuron cells, that I don't know. But if you use the same type of cells, SHSY5Y, it's definitely workable. And we recently published a work just by real-time treatment of neuroblastoma cell with alpha-synuclein, and we can see the gradual change during the process, if that answers your question.
Yeah, it did. In fact, I'm also using SHSY5S cells, and I'm particularly interested in one mutation which affects protein-protein interaction. We don't know much about that. So one effect of that protein-protein interaction is increased neuronal branching. So
I see.
Cool.
Yeah, so we can run the SICM, the morphological change experiment. We can also monitor the real-time binding of your protein to the cells if you're interested in that part.
Excellent. I will share my contact information with you, and then we can.
Thank you.
Thanks.
Great, thanks, Param. Next questions. Well, I have a question for Ornella. I'm very fascinated by your use of math and materials. Do you have limitations as to how, and I actually saw your other 3 projects there too, which are quite interesting too. Are you limited in terms of certain lattice types, crystalline, amorphous? Can you drill down a little bit more in terms of that?
Sure. So in the cases that I showed, you see that the vertical axis is time. So we're not working with a 2-dimensional crystal, but we're working with a 1-dimensional crystal. And the only restriction that you have is that the way you change the properties in time needs to happen at a speed that has some restriction compared to the speed of the wave inside the materials. Because if the two speeds are comparable, what happens is that you have shocks, or what happens is that actually the wave doesn't even propagate through because either one of the two is too faster compared to the other. So yes, there are limitations that can be proved mathematically. So it's not that every shape can work. The geometry of the material is limited by that condition, that the modulation speed has to be in a certain ratio with the wave speed in the materials. Yeah.
Great, thank you.
Thank you for the question.
So hi, this is also for Ornella. This is a little bit outside the bounds of your presentation, but I was just wondering if you could briefly share sort of what you work on in terms of viscoelastic composites. My background is, sorry, I'm Julie Simons from Cal Maritime, and I work on mathematical models for sperm motility. So I'm interested in viscoelastic fluids.
Oh, okay. So I work on viscoelastic composites, so solids. So that's like the huge difference here. But basically what I'm trying to do is to predict the behavior in time of the composites. You know that the majority of the results out there when you work with viscoelasticity are in the frequency domain. But what we're trying to do is to do it in time. And there is a reason for that, because we're interested in the opposite problem. That is, if I know the response of the composite, can I gather the volume fractions of the components? That's what we want to do, just by doing some measurements in time. Yeah, so I don't know if there could be some overlap, because viscoelastic fluids and solids are very different. Yeah.
Totally true. Yeah, thanks.
Thank you.
Sudhir, I had a question. You mentioned the use of ultra pure air in terms of a condition for some of your work. So say, for example, with these dogs, or what you're trying to mimic in your sensor, somehow dogs are able to do something that is quite interesting, quite novel, quite innovative. But in so many ways, they have an ability to discriminate between other types of VOCs and key on that one, even though there may be noise from others VOCs that are there. So do you have the ability, or your sensors you're developing, are they to the point that you can almost simulate what dogs are able to do to that extent? Because somehow in their brain, their neuronal system, what have you, they're able to, and I don't know what the LODs are either in terms of their detection limits. Do you have any information on this?
Yeah, I think, what we're trying to do is, more in a way, mimic some kind of function, because, I think it's not even 0.1% of, what dog's nose can do, right? They have hundreds of thousands of sensors, and I think there are 75% of brain is dedicated for smell, and they have the ability to collect the molecules. So, dog's nose is very, very complex. It's not even 1,000th of what dog nose can do. But I think over time, and when you look at what is in the breath, and some of the previous analysis, looking at the concentrations of the levels of these compounds, and the ability of the sensors that we have, and some of these sensors are commercially available sensors already. So that means that these have been tested, so that's the great thing about the off-the-shelf sensors. If you look at the individual sensors, they might be able to detect some of these compounds, but that won't be much of that can't give you much information. To deduce any information from the sensors, we need to have multiple sensors, and it needs to be connected with a well-trained machine learning algorithm. So I think it will be able to do that once you have enough data collected from the sensors directly from the patients when they are at the different blood glucose levels. And with that, with the machine learning tools and in good of a computing power, we have seen enough information to say that it is possible to classify them, but it won't be at the level what dogs can do.
Thank you.
Thank you for the question.
Any other questions for our speakers? If not, I certainly would like to thank Yixian, Ornella, and Sudhir for giving us some very interesting highlights of their work and potential collaborations going forward. It certainly was exciting to hear about what they're doing, and hopefully, even if we get just one potential collaboration, it's certainly worth all the effort in putting this together. Our next our next virtual research cafe is November 5th at 2 p.m. The registration will be sent out very soon. It's Monica actually put that into the chat box, but you'll also be receiving that if you are on our listserv, and if you're not, you will be very soon for attending this. I thank all of the registrants for attending, and mind you, every time we do have one of these cafes, it's a mix of faculty. We'd like to mix it up. It's kind of like having a party where you kind of invite all these different groups of people, and as long as they don't fight and beat each other up, then you actually will have a good party. So hopefully this was a good one for you to kind of take a little bit out of your day to learn a little bit about science and what some of your colleagues are doing going forward, instead of teaching or grading or whatever else you're doing. It's always nice to actually do what all of our degrees were meant to. Not that teaching and everything else that we do is not rewarding, but certainly it's always good to actually drill down in terms of the science, which I think that's part of which was why we went into the STEM disciplines. So to all of you, I say thank you. And of course, I always have to say thanks to Monica for keeping me honest and on the yellow brick road in terms of these virtual cafes. So to all of you, thanks. To our speakers, thank you. And do not hesitate to contact us at STEMnet and our other activities going forward. So have a good and safe day, and we'll be seeing you. Bye.